Hello students, today we are going to start our new topic which is mitosis. So let's get started. What is mitosis? Mitosis is that step in the cell cycle where the newly formed DNA is separated and two new cells are formed with the same number and kind of chromosomes as the parent nucleus. What are the stages of mitosis? The stages of mitosis are given below. Number 1, interphase. Number 2, prophase. Number 3, metaphase. Number 4, anaphase. Number 5, telophase. What is interphase? Before entering mitosis, a cell spends a period of its growth under interphase. It undergoes the following phases when in interphase. G1 phase. This is the period before the synthesis of DNA. S phase. This is the phase during which DNA synthesis takes place. G2 phase. This is the phase between the end of DNA synthesis and the beginning of the prophase. The next phase is prophase. Prophase immediately follows the S and G2 phases of the cycle and is marked by condensation of the genetic material to form compact mitotic chromosomes composed of two chromatids attached at the centromere. Completing the prophase is characterized by initiating the assembly of the mitotic spindle, the microtubules, and the proteinaceous components of the cytoplasm that help in the process. The nuclear envelope starts disintegrating. The next metaphase is started. At this stage, the microtubules start pulling the chromosomes with equal force and the chromosome ends up in the middle of the cell. This region is known as the metaphase plate. Thus, each cell gets an entire functioning genome. The next stage of mitosis is anaphase. The splitting of the sister chromatids marks the onset of anaphase. These sister chromatids become the chromosomes of the daughter nuclei. The chromosomes are then pulled towards the pole by the fibers attached to the kinetochores of each chromosome. The centromere of each chromosome leads at the edge while the arms trail behind it. The next and last stage of mitosis is telophase. The chromosomes that cluster at the two poles start coalescing into an undifferentiated mass, as the nuclear envelope starts forming around it. The nucleolus, Golgi bodies, and ER complex, which had disappeared after Prophase start to reappear. Telophase is followed by Cytokinesis, which denotes the division of the cytoplasm to form two daughter cells. Thus, it marks the completion of cell division. Thank you so much for listening to this video. If you have any questions, comment below. And kindly like, share, and subscribe to my channel for more informative videos. Bye!